Dignitaries, learned delegates, respected professors and students, a very good morning to one and all. I, Abhijit Govind, a sophomore pursuing biomedical engineering at the National Institute of Technology, Rourkela, consider it an utmost privilege to welcome you all to this conference on behalf of Genesys. I take this opportunity to specially welcome all the head of departments, professors, faculty members, students of various departments, and esteemed audience who have all gathered here from diverse places. There is this quote on the internet that I recently came across. It goes like this: "Be careful now that I can see the inner you." Well, with the tremendous advancements made in the field of medical imaging and the fact that it is still growing exponentially makes the above quote scary. The field of medical imaging, stimulated by advances in digital and communication technologies, has grown tremendously. New imaging techniques that reveal greater anatomical detail are available in most diagnostic radiology departments. We discuss vascular imaging with ultrasound, magnetic resonance imaging applications, and picture archiving in communication systems. Therefore, it is with great honor and with utmost privilege that I introduce our honorable speaker, Dr. Prateek Prasanna, for today's session on medical image analysis. Dr. Prateek Prasanna is an assistant professor in the Department of Biomedical Informatics at Stony Brook University and directs the Imaging Informatics for Precision Medicine Lab. He received his PhD in Biomedical Engineering from Case Western Reserve University, Ohio, USA in 2017. Before that, he obtained his master's degree in Electrical and Computer Engineering from Rutgers University in 2013 and a bachelor's degree in Electrical and Electronics Engineering from the National Institute of Technology, Calicut, India in 2010. Dr. Prasanna's research focuses on building clinically translatable machine learning tools that leverage multiple data streams of imaging, pathology, and genomics to derive actionable insights for enabling better treatment decisions. His research has involved developing companion diagnostic tools for thoracic, neuro, and breast imaging applications, and has won several innovation awards and received recognition from the Ohio Secretary of State and House of Representatives. One of the core focuses of his lab is to integrate machine-generated interfaces with expert clinical research to make clinical workflows more efficient and effective. I sincerely hope that this conference will be a great success, not only as an opportunity to share knowledge and experiences in higher education, but also as a beginning of a plethora of stimulating and interactive discussions. With that, I hand over the proceedings to Dr. Prateek. Sir, please.
Thank you so much, Abhijit, for the great introduction, and thank you for inviting me to this conference. So this is actually special to me in more than one way because this is NIT Rourkela is where I attended my my counseling for my undergrad, and that's where I ended up at NIT Calicut. So anyway, so I'll get started. So let me just share my screen. Let me know if you can view it. Just give me a second. Okay, do you see my screen?
Yes, sir, it's visible.
Okay, great. So my talk today is titled Augmented Intelligence in Medical Imaging for Precision Medicine. And as you can see here, I've purposefully struck off artificial here. And now there's a particular reason, why I have done this. And the goal of my talk today would be to put this point across to the attendees in this conference. So for those here who work in artificial intelligence and machine learning, we understand that humans and machines cannot work in a siloed fashion. So domain knowledge exists at various different levels, and wherever it exists, it should be integrated as much as possible to our inference systems. A lot of times we, get hung up with performance metrics such as, AUC, accuracy, precision, recall, etc., etc. Now, if one does not understand why a system generates a result that it does, it is difficult to adopt it for real-world use, especially when it is in the realm of medicine. Now, machine learning should not be used just to get one up on human intelligence. We should use it for hypothesis generation and to augment the intelligence of clinical experts wherever possible. So here's an article that states why AI and human intelligence must work together in the field of medicine. Now, AI isn't here to take our jobs. That has been the quote unquote the story in many leading dailies every single day, day in and day out. But we should realize that AI isn't here to take our jobs. Rather, it is here to accelerate and better inform the decisions that humans make. By marrying AI with human expertise and intuition, we can significantly improve the way medical care is provided, to patients, to make sure that the right care is provided to the right patient at the right time. Now, let's think of it as augmented intelligence to reveal new ways to leverage data, treat patients proactively, and optimize the use of available resources. Now, the slide here summarizes the broader impact, what I'll be discussing in the next 30 minutes or so. So this was an article in Data Science Central from a couple of years ago. It shows the huge gap between the current state of machine learning research and its clinical acceptance. From proof of concept to production, there's a huge gap. Now, why is that the case? Now, in this talk, I'll be focusing on some of our efforts towards bridging this gap. This article states here that doctors are from Venus, data scientists are from Mars. Why is AI ML moving so slowly? For those of you who work in computer vision or machine learning, the state of research in machine learning is, let's say, here, and what we have, what is currently being implemented in the clinic is somewhere down here. Now, why is there such a huge gap? So some of the tools and techniques that we have been developing as part of the research that we conduct here has been to address this particular gap in the field of medical vision. So Yoshua Bengio, in one of his interviews, mentioned that deep learning is fundamentally blind to cause and effect. Now, unlike a real doctor, a deep learning algorithm cannot explain why a particular image, for example, may suggest disease. Now, this means that deep learning must be used cautiously in critical situations, especially in situations where, where the life of a patient, is on the line. We cannot really trust systems unless we understand why a system makes a particular decision that it has actually made. So this brings the aspect of intuitiveness and interpretability, which are probably the most important aspects now in AI in medicine when clinicians are tasked with making high-stakes decisions. Here's another article in The Wired that says AI can help patients, but only if doctors understand it. Well, not everyone might agree with this, but the fact of the matter is that there is certainly, a degree of truth to this. It again comes back to the aspect of interpretability and intuitiveness of the different approaches that we develop. There can also be other challenges that clinicians should be wary of, and rightfully so, as shown in the New York Times article right here that says, warnings of a dark side to AI in healthcare. One such particular aspect is adversarial attacks, such as, altering a few pixels of an image that can actually lead to change in diagnosis, for example. Now, to address the issue of interpretability, my talk today will be focused on biologically inspired and domain-inspired tools that we have been working on and how we may be able to use such tools to improve the intuitiveness and interpretability of machine learning. Now, heterogeneity is observed at many different length scales in the field of medicine, such as the gene level, the cellular level, the histologic level. And we believe that the heterogeneity in the lower length scales is also manifested on imaging and can be captured via different kinds of imaging techniques. Now, imaging provides a complementary landscape of characterizing the underlying heterogeneity orthogonal to what may be gleaned from the different are the length scales of data. Now, it is important to characterize heterogeneity since it is associated with poor prognosis and treatment response in the context of several different treatments. Now, in order to quantitatively characterize the underlying disease heterogeneity, we have been working on the development and validation of different tools such as radiomics, Pathomix, and radiogenomics biomarkers. Now, these approaches have been developed across domains, including brain, breast, and lung cancer, in the context of disease diagnosis, prognosis, and treatment response evaluation. Now, the research theme of my lab, it lies at the intersection of machine learning and image analysis, where our focus is on biomarker discovery via machine learning and, more importantly, multi-scale data fusion. Now, there are four fundamental questions that we seek to address through our research. What is it that we do? Our goal is to interrogate heterogeneity on multiple different length scales. Why do we want to do this? So we know that heterogeneity has been linked to treatment response and eventual outcome. So it is important to understand this heterogeneity from multiple different levels. Now, where do we look at in order to understand this? We specifically focus on the lesion habitat and the peritumoral regions as well. And how do we do this? We do this via different approaches such as radiomics and Pathomix, which I will discuss in detail going forward. So over the last few years, we have shown the utility of quantitative imaging biomarkers in diagnostic and prognostic applications, and more recently in predictive applications such as determining response to certain kinds of treatments such as immunotherapy and chemotherapy. Now, these quantitative imaging biomarkers include, features such as textural radiomics, deformation-based radiomics, vasculature radiomics, and quantitative histomorphometric descriptors. Now, before I go into some of the application domains, I'd like to spend a few minutes on explaining what these different tools and techniques are. Now, first, coming to radiomics, we have different kinds of radiological modalities such as MRIs, CTs, chest X-rays, and so on and so forth. So when we have the scans available, we can extract different kinds of subvisual features which may not be discernible, to the human eye. So we can extract features such as shapes of the different constructs here. We can extract features like texture. And these can be used in conjunction with different machine learning techniques, be it unsupervised machine learning techniques such as clustering or supervised learning techniques, including different kinds of classifiers. And this can be used for generating better informed decisions. So this brings up the question, can we develop these quantitative imaging biomarkers that are that give us a subvisual appreciation of the disease characteristics on imaging to characterize the underlying heterogeneity and aid in precision medicine? Now, here I'd like to briefly introduce the field of Pathomix as well. So similar to radiomics, where you extract subvisual features from the radiological imaging modality, Pathomix features help us appreciate the subvisual characteristics of digitized pathology slides. Now, this is on the left, the entire screen, what you see here, this is an example of a digitized H&E slide. So you have the H&E marker, which the slide has been stained with, tissue has been stained with. And so this is what a pathologist would see when they look at the slide under the microscope. Now, this would, qualitatively, if you look at an image like this, you can see, there are different kinds of cells here, and there's a particular pattern in which these cells are organized. But can we mathematically quantify these kinds of patterns that you see in an image? Now, as it turns out, yes, we can. So we can use image analysis, we can use machine learning techniques to provide a more quantitative interpretation of what this image actually looks like. So we can mine different kinds of patterns, and then this can be used to to study the nature of different diseases. So a simple step that can be done here is extracting or segmenting out the nuclei that you see in these images, and this can be done using different kinds of algorithms, be it simple image analysis algorithms or deep learning algorithms. So once you extract the nuclei, you can use the nuclei as let's say the nodes to as nodes to construct graphs. And once you construct these graphs, you can look at this, you can study the spatial proximity of the nuclei with respect to each other and the spatial proximity of different groups of nuclei with respect to each other. So that gives you one level of quantitative information. You can also use other kinds of image analysis techniques to extract different patterns such as textural patterns or different kinds of intensity based patterns and see how these patterns are different in different regions of these images. Now this can tell us a lot more about these images as compared to just a visual diagnosis. So coming to a few application domains where these tools and techniques have been found to be very useful. So I'll start with, in fact, I wouldn't be doing justice to this talk if I don't start with applications related to COVID-19. So during the beginning of the pandemic, a big question was, when we really didn't have that, that much resources, we were struggling to allocate resources intelligently to areas where there was scarcity of resources. So we thought, how about we use something that is abundantly available, such as chest radiographs, to predict the requirement of mechanical ventilation and mortality in COVID-19. Now, the unmet need here was to see can these baseline chest radiographs or CXRs provide information regarding disease progression. So in order to address this, we did a multi-site study. This was a study conducted at Stony Brook and Newark Beth Israel Medical Center, which is part of Rutgers University. In the initial part of the study, we had about 300 cases from Stony Brook and 67 from Rutgers. Now we have expanded it to more than 2,000 images, but this was during the first few months of the pandemic. So we trained machine learning models using baseline CXR scans. Now these scans were collected when the patients presented with COVID-19. And so we used different kinds of tools and techniques. We used deep learning techniques as well. And our results suggested that, yes, we can use baseline chest radiographs to predict the need for mechanical ventilation with a sensitivity of 90% and a specificity of 71%. And this was on the test set of 67 studies from Rutgers. So, however, what was more interesting was to see if we could use imaging to provide evidence of disease improvement on sequential data. So none of the previous works, jointly exploited the spatial distribution within the images and the temporal distribution across the time points. Now, what do I mean by this? So if you look at this series of images, you can see that, you find these lung infiltrates, these chest infiltrates here. This is on day one of the patient presenting with COVID-19. And we can see how the infiltrates become denser as you progress to day 2 and day 3. And eventually the infiltrates resolve and it becomes similar to day 1, and then it becomes better going forward. Now, can we use imaging? Let's say, can we use images from day 1 and day 2 to predict what will happen on day 3 and what will happen on day 4? So that was our goal for the next part of this project. Now, for that, we used a sequential chest X-ray data set with about 600 scans from close to 100 COVID-19 patients. And then radiologists looked at these images and assigned severity scores ranging from 0, 1, 2 to the different lung zones. Now, what we first did was we divided the lungs into different zones. And for this, we needed to first develop a deep learning algorithm to segment out the lungs. And once we had done that, we divided into the upper lung, middle lung, and the lower lung which was assigned a grade of 0, 1, 2, depending on the level of the severity of the lung infiltrates by the different radiologists in our study. Now, the goal was to use images from the first few days and predict the severity of the disease in the different zones of the lung on let's say the nth day. Now, in order to do that, we developed this spatio-temporal CNN LSTM architecture where we had a temporal LSTM block and a spatial LSTM block. Now, the spatial LSTM block looked at or it quantified the relationship between the lung infiltrates in the different zones of the lung on a given day. And the temporal LSTM block looked at how the infiltrates evolved across the different days. And once we did that, we found that we could indeed predict what would be the severity of the lung on the nth day, depending on the first on the images of the first few days that was acquired in the first few days. And we compared our approach against different radiomics based approach and other variants where we removed either the spatial block and the temporal block. And we found that incorporating both the spatial and the temporal block helped us predict the severity with a better performance with better performance metrics. Now, coming back to the first couple slides of what I discussed, how do we integrate domain knowledge into a machine learning framework? Now, when radiologists look at these images, I mean, when you look at the progression pattern of the disease from day 1 to day 2 and so on, you see that the infiltrates progress in a certain characteristic way within the lung. So typically they start out in the lower lung regions and they slowly move up to the upper regions by traversing the middle region. I mean, there might be exceptions, but this is typically the case when it comes to the progression pattern of COVID-19. Now, this helped us, design our system to better understand or to better incorporate this domain knowledge and better predict how the progression would look like, n days from now. So what we did was we introduced this correlation module in our network. Now, what this correlation module does, I mean, to simplify it, it looks at the, if I go back one slide, if you look at the correlation between the disease patterns between, let's say, the neighboring zones. So the lower zone and the middle zone can be considered as neighboring zones, and the lower zone and the upper zone can be considered as far away zones. So the correlation of the disease patterns between adjacent zones would be much higher as compared to the correlation of the disease patterns in the further away zones. Now, this appears to be a very simple concept, but integrating this kind of a module within our deep learning network helped us improve our performance of prediction of severity in the final classification approach. So this helped us increase both our precision recall and accuracy for for progression prediction. So now, one big question when it comes to developing machine learning or let's say deep learning approaches in this field of medical sciences, dealing with limited data scenarios. Now, when this pandemic started, obviously we did not have any idea about what a COVID image would look like. Now, gradually images started flowing in from different places. First, they started coming in from places like China, where the a pandemic first originated, and gradually we started seeing cases from other places in the world. However, when you do not have images, let's say in the first few days of the pandemic, or when you have limited imaging data, can you still use these images to study how this disease might progress or to predict what the eventual outcome of patients will be? Well, yes, we might be able to do so using handcrafted feature engineering, but the results that you get might not always be good. So it's always good to have more data. However, how do you how do you use the limited data to generate something that you can use to develop data hungry algorithms like deep learning techniques. So the next work that I'll talk about addresses this question. So so this work actually is based upon the hypothesis that the texture of a disease is something that is related to the eventual long-term outcome. So here you can see a patient on day 2. This is a COVID patient on day 2, and you can see how the infiltrates aren't very dense. However, on day 23, you can see that the lung infiltrates present a very dense pattern here. This is actually shown in a zoomed in version right here. You can see on day 2, you don't really see much infiltrates, and on day 23, you can see a high concentration of these infiltrates. However, interestingly, if you look at the shape of the lung here, you see that the shape of the lung on day 2 and day 23 doesn't really change much. So the shape of the lung hasn't changed, but the texture within the lung parenchyma has changed a lot. So what this tells us is that the in-lung texture correlates with infiltrate levels, while the lung structure remains unchanged throughout the disease course of an individual patient. Now, we can use this observation to develop techniques that would help us, create new X-rays that preserve lung structure and can introduce new texture to these lungs. And this will be clear in the next couple of slides. So the observation here was that in-lung texture is reflective of disease severity. Now, we developed this new technique. It's called the lung swapping autoencoder. This is a representation learning based technique. So to simplify this architecture that we have here, we develop a structure encoder, which essentially encodes for the structure of the lung. And we have a texture encoder, which encodes for the texture of the lung. Now, using the structure encoder and the texture encoder, we learn the different patterns relating to the structure and the texture of the lung, which can then be used to generate artificial or synthetic samples of COVID X-rays. Now, here's an example of the lung swapping results that we obtained. So with both in-lung texture supervision and out-of-lung distortion suppression, we can achieve reasonable lung swapping results, as you'll see now. So the first image, you can see the structure of the lung here, and the second image, you can see the texture of the lung. So the second image is actually an example of a healthy lung. You don't really see any infiltrates here. However, in the first image, you can see certain infiltrates. Now, when we use our method and we develop this hybrid lung image, we can see that we have clearly retained the structure from lung 1, and we have artificially introduced the texture from lung 2. So you can see how we have been able to successfully remove the disease patterns in lung 1 through the texture encoder from lung 2. Now, similarly, you can also use the structure of lung 2. So this image in the lower part, you can see the structure of lung 2. And we have been able to artificially introduce the texture from lung 1. So you can see some of the COVID patterns right here. So we, in fact, conducted an experiment where we showed these images to radiologists, and we saw that in about 75 to 80% of the time, radiologists couldn't really say if this was taken from a real patient or if this was an artificially generated image. Now, we can clearly see how a technique like this can help us address limited data scenarios. Now, if we can augment our data set through this domain-inspired method, we can use this to train data-hungry algorithms. Now, after our lung swapping autoencoder was trained, we applied it to perform two downstream tasks. First, we used the texture encoder in the lung swapping autoencoder to do pulmonary disease classification on a large scale, chest X-ray 14 data set, which had close to 112,000 images. And we found that our texture encoder was comparable to other algorithms such as Inception v3. However, our encoder, the number of parameters was a lot lower as compared to the parameters used in the other algorithms with a similar AUC performance. The second task was a COVID-19 outcome prediction on a COVOC dataset, which was curated locally at our institution at Stony Brook. So compared to a native SAE, our method LSAE could achieve a superior performance, which demonstrates that a better disentanglement leads to a better performance, as you can see here. Now, coming to the augmentation task, we used the swapping results that I just showed a couple slides ago to artificially augment our dataset 2 times. And we saw that once we had augmented our dataset 2 times, our performance increased by almost 1.5 points, as you can see here. Here are some qualitative results that we obtained. So the top first 3 rows are examples of correctly classified cases, and the bottom row are examples of misclassified cases. So red regions are essentially the regions where the COVID infiltrates are, and these were marked by our expert radiologists. And these are class activation maps. Class activation maps, as some of you may know, basically looks at the regions or provides information regarding which regions in the image the algorithm focuses its attention on to give you its results. So we can see that for the most part, the algorithm focuses on regions where there are COVID infiltrates. However, there are cases of misclassification as well, as you can see here. So I'd like to switch gears a bit and talk about some of the applications in the field of oncology. Now, the first clinical challenge that I would like to discuss in the field of oncology is distinguishing radiation-induced effects from recurrent tumors in brain cancer. So just a quick overview of the treatment paradigm and what the unmet clinical need is. So once a brain tumor patient has undergone surgery, they would undergo radiation treatment along with chemotherapy. Now, as the treatment progresses, the patients, they usually come back for follow-up imaging, and there might be suspicious findings on follow-up MRIs, as you can see here. Now, these suspicious findings, they might either be, the lung tumor, the brain tumor that has come back, or it might just be a benign effect of radiation. Now, if these, changes appear within the first 3 months, there might be something called pseudoprogression, or if they appear after 6 months, they can be radiation necrosis. So pseudoprogression and radiation necrosis are tumor mimicking effects. They are not tumors. So, however, they look very similar to recurrent tumors, and that's why they're called tumor mimicking. Now, if they're pseudoprogression or radiation necrosis, the treatment is less aggressive, and more often than not, it's a simple MRI follow-up. And if it is a recurrent disease, there probably needs to be another resection, another biopsy, or change of treatment. However, it is very difficult to distinguish these conditions. Therefore, surgical intervention is the only foolproof way of definitive diagnosis. So, by looking at images, radiologists can distinguish between these 2 conditions. However, with the standard imaging protocols that are out there, T1, T2, and FLAIR, the performance is probably around 55 to 60%. So we asked ourselves the question: can we use treatment naive imaging, right after surgery has been done, or right after chemotherapy has been done, to predict whether what you see on the image is either radiation necrosis or recurrent tumor? So a few years ago, we developed an orientation entropy-based descriptor. So this is a collage descriptor. So these collage features are unique in the sense that they capture the co-occurrence of dominant orientations at pixel level, and they bring together the classical GLCM-based texture features and steerable filters by capturing statistics and frequencies of local gradient orientations. Now I'll explain what I mean by that. Now, what you can see on the screen right now are examples of, so one of them is a radiation necrosis and the other one is a recurrent tumor. Now, radiation necrosis is a relatively benign condition, while recurrent tumor is a more aggressive condition. Now, maybe let's play a pop quiz here. So can any of you here tell me which of these is radiation necrosis and which of these is recurrent tumor? And if it's difficult to, make that call, how about now? Which do you think is radiation necrosis, or which do you think is a more benign condition, and which do you think is a more aggressive condition?
Sir, I think pathology 2 is the more benign condition.
That's right. And why do you think that's the case?
Because since
So
the
what
tumor
led you to
is
that
being
answer? Yeah.
radiation cannot pass.
Sorry, I didn't get you. Sorry.
Since the tumor is formed, the radiations are being obstructed by the tumor.
Okay, so as you can see here from the entropy map, we can see there are more cold spots here in the right example in the image on the right as compared to the image on the left, and right one is indeed an example of benign radiation effect, and the left one is an example of recurrent tumor. Now, collage feature, in fact, this is a concept that we borrowed from high school physics. We look at the entropy of the different gradient orientations, and we found that the entropy is obviously higher for the recurrent tumors, and the entropy is lower for radiation necrosis. So what this tells you is there are changes on the radiologic length scale that can be mined using image analysis and machine learning approaches. And this can essentially tell you what's going on at the pathologic or the cellular length scale. So what we also did was we took examples or we took cases with different grades of radiation necrosis and tumors. So on the left, you see a case of nasopharyngeal carcinoma. So this is a pure radiation necrosis case. And on the right is an example of a glioblastoma multiforme. It's the most aggressive kind of brain tumor, and no radiation has been given to this patient. So this is an example of pure tumor. And the images in the middle that you see here are examples of mixed tumor and necrosis. So this one has higher necrosis, and the one on the right has higher tumor. So when we extract our collage features, we see that there is a clear gradation of these features. Just give me a second. My computer is acting up. I'll just restart my PowerPoint. Just give me a second. Okay, can you see my slide now?
Yes, sir.
Yes.
Okay, yes, sorry about that. So yeah, so what we did was we extracted our features, and we find that there is a clear gradation of these features going from pure necrosis to pure tumor. So this kind of change you cannot really see using the, just based off the original images, but when you extract these collage features, we can appreciate this change. Okay. Okay, so now, obviously the proof of the pudding is in the eating. So how do we know that this feature actually can be used in clinical decision making? So we had a test set of about 15 cases, and then we asked two independent readers. So we didn't, so they weren't provided any information regarding the ground truth, and they were asked to independently grade these cases into either the category of radiation necrosis or recurrent tumor. And then we had a radiomics classifier as well. So we found that the tumor detection accuracy of the radiomics classifier was a lot better than compared to that of the individual readers. Now, more importantly, when we combined the decision of the radiologist and the radiomics classifier, the accuracy increased even further. So this shows that tools like this can be used to augment the decision-making capabilities of radiologists. So one case that was misdiagnosed by both the experts and the classifier was a metastatic recurrence case. So the second challenge in the field of brain tumor management is predicting clinical outcomes on treatment-naive imaging. So now there are close to 15,000 deaths every year from patients suffering from GBM. Only about 5 to 10% patients have good outcome, which is greater than 3 years of survival. So understanding differences in survival characteristics can allow for designing personalized therapeutic trials. So the current treatment approach is a one-dose-fit-all approach. Tumor heterogeneity on treatment-naive MRI has been linked to poor prognosis. Therefore, there's an unmet need to develop techniques to quantify this heterogeneity. Previous groups had looked at different kinds of approaches, such as shape-based approaches, volumetric approaches, coming from the different regions of the brain tumor. So we took a more nuanced approach where we looked at not just the regions inside the tumor, but also the regions surrounding the tumor. I'll talk about it in the next few slides. So here's an example of a patient who survived for less than 8 months. And here's an example of a patient who survived for greater than 18 months. Now here's the tumor habitat as annotated by a radiologist. The yellow region is the enhancing tumor. The green region is the inner necrotic core, and the brown region is the peritumoral edema. Now, typically, when radiologists look at these images, they would first, look at the enhancing tumor region, then probably the necrotic core. But the decision is not really based off what happens outside, the tumor core. However, when we extracted our radiomic features, we found that there was a clear difference in the expression level of these features, even in the outer peritumoral zone. Now we can see that there's a higher expression of these features in the peritumoral zone, also in the inner necrotic core, as compared to the contrast enhancing zone. So what this tells you is there is, a prognostic signal, not just inside the tumor, but also outside the tumor, which can be appreciated using different, mathematical descriptors of imaging heterogeneity. Now we use these features in conjunction with survival analysis algorithms. So these are Kaplan-Meier survival curves, and we can see that combining radiomic features with clinical features gave us the best concordance index. So next, we hypothesized that tissue perturbations due to mass effect, it leads to heterogeneity in normal parenchyma and can be captured using measures of extratumoral deformation. Now, what this means is, because of the peritumoral edema, which is essentially the fluid which surrounds the tumor core, so the fluid sort of exerts pressure on the healthy parenchyma, and we hypothesized that the pressure that is exerted by this fluid, it results in deformation of the healthy parenchyma, which is different between patients who survive longer versus patients who survive shorter. So what we did was we registered these images to a healthy atlas, and we computed voxel-wise deformation magnitudes. And we can see that for a short-term survivor, the deformation is much higher in the regions surrounding the tumor as compared to the long-term survivor. Interestingly, the textural patterns within the tumor were also different across the short-term and the long-term survivors. So what we also found was that when we looked at the heterogeneity of this deformation at the very edges of the tumor, we found that the heterogeneity was much higher in the short-term survivor as compared to the long-term survivor. And you can actually appreciate that in this particular video. So we can see that for the short-term survivor in the different regions of the brain, the deformation magnitude is much higher as compared to the deformation magnitude in the long-term survivor. So this was a patient who survived for less than 240 days, and this was a patient who eventually survived for more than 18 months. Okay, so now I'd like to talk a little bit about other kinds of descriptors such as Pathomix descriptors, which can then be combined with radiomics as well. So there's a lack of biomarkers currently to identify which lung cancer patients stand to receive benefit from immunotherapy. So there's a lack of objective methods to assess immunological benefit from different IO agents, so that patients who are not responding can have the treatment changed or modified. Now, here's an example of a patient who eventually did not respond to immunotherapy. And here's an example. The second column is an example of patient who eventually responded to immunotherapy. And as we can clearly see, as a result of the immunotherapy treatment, the radiomic patterns changed differentially for the non-responder as compared to the responder. Now on the right part of the screen, you see the lymphocytic infiltration patterns. So we used a deep learning algorithm to extract lymphocytes, and then we looked at the arrangement of these lymphocytes. Now the lymphocytes were found to be differentially arranged for the responders as compared to the non-responders. Okay, just give me a second. Looks like animation isn't playing here. Okay, so coming back to the same slide, so we also looked at a consensus clustering plot, a clustering plot where we found that patients who did not respond to immunotherapy had a more sparser arrangement of the lymphocytes as compared to patients who responded to immunotherapy. Now, another important component in developing biomarkers that is often ignored, where topology and geometry can play a role, is the tumor-associated vasculature. Now, angiogenesis is the process by which a tumor hijacks the body's machinery for creating new vasculature. It's a very important hallmark of cancer. So tumor-induced angiogenesis is one of the most important mechanisms by means of which a tumor adapts to changes in nutrient requirements. Now, the associated tortuosity and leakiness, they directly affect disease progression and possibly its response to therapeutic interventions. So here's an example of two patients. The first patient responded to neoadjuvant chemotherapy, and the second patient did not respond to neoadjuvant chemotherapy, and we can clearly see that the patterns of vasculature is very different across the two different patients. Now, coming back to the problem of immunotherapy that I just discussed a couple slides ago, the two most important questions are who to treat and should the treatment be continued. Now, what we did was we took treatment-naive CT scans. We used a deep learning algorithm to extract the different, the vasculature that feeds the tumor. And then when we used measures of tortuosity, we found that the vessels that fed the tumor for a responder were the vessels were much less tortuous and much less convoluted as compared to the vessels that feed the tumor in a non-responder. Now, borrowing the analogy from, let's say, electrical engineering, what this possibly points to is the fact that because of the lower tortuosity in the responder, there might be a path of least resistance for the drug to affect the tumor as compared to the non-responder, where because of the more tortuous nature, the drugs are not able to attack the tumor as they are originally intended to. Now, what we also saw was when we looked at the tortuosity change between the baseline and the post-treatment scans, the responders to this particular treatment, as a result of the treatment, the tortuosity actually went down for the responders. However, in the non-responders, even after the treatment, the tortuosity did not go down, or in some conditions, they actually went up. Now, finally, here's an example of a histomorphometric feature that can be used to quantify, the interaction between different groups of nuclei. So this was a project where we sought to evaluate the differences, or we sought to predict patients with long-term survival and short-term survival in non-small cell lung cancer. So we first segmented the nuclei and then used a clustering technique to identify local clusters of the nuclei. And then we looked at measures such as the area in the convex hull and the intersection between the areas of the different groups that we had identified. And we found that using these handcrafted features, we could predict prognosticate the survival in lung cancer patients. In fact, our handcrafted features were found to be much better than some of the standard deep learning features here. So here's a qualitative example of, so like I said in the beginning, it's always important to understand what these features mean and show these features to clinicians and see if it actually means something from a visual perspective. Now, if you look at these high-risk patients and the low-risk patients, so we had so the blue cells are the cancer nuclei, and the yellow cells are the tumor infiltrating lymphocytes. So we can see that the arrangement of the cancer nuclei with respect to the tumor infiltrating lymphocytes are very different for the high risk case as compared to the low risk case. Okay, so the final application that I'd like to discuss here is, again, how do we augment a radiologist's decision making by looking at their eye gaze information? So there's a certain characteristic pattern in which radiologists look at these images, be it MRI scans, be it CT scans, be it chest radiographs. So there's a certain way in which they are trained to look at these images. Now, can we use machine learning or deep learning techniques to first study how radiologists look at these particular images, and then after understanding the inner mechanism of how the gaze patterns work, can we use that information within another machine learning algorithm to improve the performance of the second algorithm? So this is something that we did very recently. This is under review right now. So what we call this here is a human visual attention training. So we get the eye gaze points using a gaze tracker. And then we have a teacher model, a global focal teacher model that learns how these radiologists look at these images. So once this teacher model has learned how to understand the gaze patterns, it teaches the student model to replicate the pattern using the predicted attention that you see here. And now we develop a visual attention loss that is then used to improve the this automated disease classification task. Now, the way we design this is using global blocks and focal blocks. Now, the reason we design these global blocks and focal blocks is to mimic the way radiologists look at images. Now, focal block essentially looks at focal changes or very local changes within the images, coming from one pixel to another in the very same region or in its immediate vicinity. However, global block looks at the overall patterns in these images. So the way radiologists look at these images is first they form an impression of what disease there might be in a given image by looking at it in a zoomed out version, and then they zoom in into these images to look at the localized changes. So our deep learning algorithm does exactly the same. It first has a global block, and then it has a focal block, and the global information and the focal information, they feed into each other to make sure that both these information are combined to arrive at better decisions. So we use this technique to predict or to diagnose pneumonia and regions of pneumonia and COVID-19. And we found that the regions that our algorithm showed was it overlapped well with what the radiologists found out. So finally, I'd like to finish with this particular note saying that, augmented intelligence is indeed the next frontier in health imaging. Unlike artificial intelligence, which tries to replicate human intelligence, augmented intelligence works with and amplifies human intelligence. So here's a funny Twitter conversation that was going on between certain experts. In fact, a couple of them are actually radiologists. So where you'd have probably heard this narrative a lot of times where people say radiologists who use AI will replace those who don't. Well, the fact of the matter is that that's not the case, and that's for multiple different regions, and nothing more than the fact that we don't have enough radiologists. So there's no way that radiologists are replacing other radiologists. What is more important to understand is that radiologists who understand AI will augment those radiologists who do not understand AI, and that's the way the field is headed today. So I'd like to thank all my students at Imagine Lab, all my collaborators both at Stony Brook and elsewhere, and all our funding agencies. So that's all that I had today. I'm happy to take any questions.
Hello. All the participants are requested to put their questions in the chat box, and even they can unmute and ask.
Okay, so I see raise of hand. So Sharat, yeah, please go ahead.
Yes, sir. Good morning, sir. So actually, like you talked about augmented intelligence. Though I don't have like much prior knowledge in this, but what I know is like both artificial intelligence and augmented intelligence, they like both of them work on machine learning, but like I think the procedure is somewhat different. So can you explain that a bit?
So that's a good question. For someone who doesn't work with machine learning, it might come across as they are two unrelated things, but they are really not. So augmented intelligence is essentially artificial intelligence that is used to augment the decision-making capabilities of classifier, for example. So the way this should be used is to not just get the best results, but to understand how a particular decision or why a particular result is being arrived at. So that essentially is what augmented intelligence. It's not really different from artificial intelligence. It's the context in which we use artificial intelligence. Hey Mirza, I see you've raised your hand.
Yes, Prateek, thanks for the very interesting and detailed talk. Actually, I just have, I think when you started off your talk with saying that it is important for us to understand how AI works. So just, I think that's a very interesting thought, because if we do not understand where AI works, then people will not have the trust to employ AI in critical situations like medical applications or diagnosing COVID or like cancer, whether it is harmful or not. So anyway, whether it is benign or not. So my question is, given the use of neural networks in these sort of applications, neural networks itself, they have some level of abstraction associated with them. So, do you know of any work, recent work, where where the effort has been to kind of explain how neural networks do prediction, or how neural networks work Do you know of any recent work in that regard? So that it will, because this is important as bioengineers, because we sit right at the interface between medical science and engineering. So I feel this is very important to understand. Yeah, over to you.
Right. Yeah. So that's a great question, Mirza. And first, thanks a lot for inviting me to this forum. And yes, so like you rightly pointed out, so we work at the intersection of medicine and engineering. So it's important for us as scientists to make sure that the tools and techniques that we develop are transparent enough for our clinical collaborators to be uh, to trust these methods enough to use them in their day-to-day practice. Now, to answer your question, whether there are recent works that have tried to address this gap, yes, in fact, I think trustworthy AI, interpretable AI is a big field of research nowadays. So one of the slides that I showed in my talk today, so I discussed about class activation maps. So that in fact is a post hoc way of explaining why or how a machine arrives at a particular decision. So what that technique essentially does is, so after a decision has been rendered, you can back propagate the loss, and you can essentially look at what were the regions in these images that the machine says or bases its decision upon. So that's just one way of understanding how a particular decision has been arrived at, and if the regions actually correspond to the actual disease regions, that does mean that the machine has learned something from these disease regions. Now this is very important because so you pointed out this fact that neural networks, there's a certain level of abstraction involved. So I think 2 to 3 years ago, there was this very classic paper that showed a machine could clearly distinguish between huskies and wolves. Now,
Yeah.
when we looked at and this was with an accuracy of 99% or even more than 99%. However, when researchers started investigating this, because huskies and wolves, they look so similar, right? As in, when we look at let's say when a child looks at a husky or a wolf, they would probably not be able to distinguish what a husky is and what a wolf looks like. So what actually happened when they looked at the inner working of the deep learning algorithm was that the algorithm was focusing on the background and not the animal. So because wolves are usually in snowy conditions where the background might be either in the forest or there might be snow. So the algorithm was actually focusing on the background rather than the animal. So these are the kinds of mistakes that can easily creep into a deep learning algorithm. And if this creeps into into the context of medical science, the lives are at stake here. So yes, so there have been a lot of work that has been going on, both for post hoc interpretability as well as interpretability while the networks are being constructed.
Okay, thanks.
Yep.
Yeah, over to anyone else.
So there is actually a question in the chat box. How the AlphaFold developed by DeepMind is helping in medical diagnosis? And are there any specific algorithms developed for studying organs using AI?
So I'm not really aware of how or what the inner algorithms the working of the inner algorithms of the AlphaGo system, and I'm not sure if any of these are actually being used in the context of medical imaging. To answer your second questions, whether there are algorithms that are being used in the context of different organs, yes. Recently, in the past 2 to 3 years, in fact, FDA has been approving a lot of AI-based algorithms. So one of the first algorithms that was approved is for breast cancer diagnosis. So this was based off of a texture-based analysis system where DC MRI scans were used, and then textures of the different lesions were computed to come up with a risk score for the different patients, whether whether the breast lesion is benign or malignant. So yes, there are different organ specific algorithms that are out there in the market now.
Just Prateek, just some interesting thought came into my mind while you were mentioning this. Since again in engineering we are we belong to the applied domain of sciences. So can you highlight any companies or startups which are actually working in this domain, so that will be of interest to the students, that will open up opportunities for them hopefully. So my question specifically is, can you please highlight any specific companies which work in the domain of using deep learning or image processing for diagnosing for medical application?
Sure. Yeah. So in India, I am aware of a couple companies, and one of them is Cure.ai, Qure.ai. I believe it's a Bangalore-based startup. I think they started in 2016 or something, and they have a line of products. In fact, maybe one of them is FDA approved, but a lot of these companies are slowly getting their products approved by the FDA and by the EU. And after that, obviously, the Indian regulatory agencies as well. But yeah, so to answer your question, in India, I'm aware of Cure.ai. Mahajan Imaging is another a group that is actively pursuing validation of AI algorithms. I think it's based out of Delhi. In the US, we have a couple startups here, Tempus. Then we have Page.ai. So Page.ai is it's a digital pathology based startup out of New York, and a Tempus, it's based out of Bay Area. So Tempus is essentially a company that deals with profiling of images and genomic sequencing as well, and how to combine imaging-based signatures with gene-based signatures to come up with recommendations for patients. It's like these DNA-based startups that have come up that look at your profile and see, okay, you should be eating healthy, you should be taking this much into your nutrients and all of that. So I think Tempus is trying to do that. So yeah, there are a few. So Philips and GE, they have their own systems that they are starting to slowly incorporate. So they have their own AI systems that they're slowly starting to incorporate into the acquisition platforms. For example, MR fingerprinting. It's a relatively new technique. This was in fact developed at Case Western. So this is a quantitative MR technique, and they have licensed it to both GE and I believe Philips right now. So essentially what happens is, once the images are taken, so they build a dictionary, and then this dictionary essentially points to if the signal expression of certain region is X, then it means it has a Y% chance of being cancer or being benign. So it's a more quantitative imaging, MR-based imaging platform. And again, that's currently being rolled out in the clinic as well. So yeah, slowly both the big players and startups are beginning to flood the market with AI-based solutions. And I can definitely send a note to the organizers with other companies that I'm aware of that do this. I mean, there are plenty right now.
Okay, great. Thanks. Over to Abhijit.
Thank you, sir. So there are a few more questions in the chat box.
Sure.
So one question is that some studies show that AI techniques are a bit unstable for medical imaging, as the small changes in the input may cause higher changes in the output. So how do we overcome these challenges to make image reconstruction more accurate?
That's a fantastic question. So in fact, yes, a lot of these techniques, especially a lot of generative modeling, which are GAN based, for example, they are very sensitive. With slight changes as input or with introduction of noise, the fidelity of the output, it changes a lot. So how do you address this? So the first way of making sure that you have incorporated all of this in your training setup is to perform enough ablation studies. So in your ablation studies, you don't just control the let's say, for example, class imbalance, but you can introduce different levels of noise to your system, and then as and when you fine tune your developed algorithm, you make sure that when you conduct these ablation experiments, the different factors that you control for do not really affect your algorithms a lot. So that's one way of making sure that this has been taken care of. So the other way which the other method that typically is ignored is that when you have your validation set, you should make sure that the validation set is actually representative of the true population, for example. So a lot of times, for example, it has been found that, let's say, methods that have been developed on a particular kind of population, let's say, Caucasians or African Americans or Indians, for example, they do not work on other kinds of population. And why is that the case? Because your training set does not comprise enough variability. So you make sure that your training set has enough variability so that when it comes to the test set, when your algorithm actually sees data in the real world, it has already seen something like that in its training set so that it can be generalizable to the real world. So there are many different ways of addressing this. I mean, these are all baby steps that must be taken in order to make sure that your system actually works in the real world.
Thank you, sir. Another question which we have is the biggest challenge with LSTM, GRUs, and other deep learning models is data collection. So how do we counter these problems?
So if was a question relating to data collection?
Yes, sir. Deep learning models, basically.
Okay.
Like the biggest challenge with deep learning models is data collection.
Okay.
So how do
okay.
we counter?
Yeah, so that's a good question. So one way that we address this challenge is, so yes, if we had, let's say, only 100 images, for example, or 100 patients in our data set, it's very difficult to design an LSTM approach, right? So what we did was, so yes, we had 100 images or 100 patients. Each of these patients had about 6 to 7 time points. So that's how we ended up with 600 images. And what we did was we took a more domain inspired approach where we divided our images into 6 different zones. Now, for each image, we had 6 different zones. So which was, the lower lung, the middle lung, the upper lung, and you have the left lung and the right lung. So instead of having just one image, we ended up with 6 different zones in that particular image. Further, instead of taking each zone as, let's say, a patch in our CNN approach, we further patched it up into 9 different patches. So this is just one way of how you can augment your data set with the limited training examples that you might have. And obviously, there are other standard techniques that are used in deep learning approaches as well. And the other thing that I also showed, the augmentation approach, where we used a lung swapping autoencoder. So we can use similar techniques to artificially augment our existing data set with similar looking images. I mean, obviously, there will be this criticism that these images may not, actually reflect the true disease characteristics. But when you use such methods, we need to make sure that these have been, or the results have been qualitatively verified by experts, and by that I mean expert radiologists or pathologists, before using these images in deep learning algorithms. Yeah.
Thank you, sir. So another question which we have is, so models have a high tendency to rely on superficial statistical patterns of the data and are not immune to the distributional shift caused by external factors. So what filters or image processing techniques can be used to reduce the noise in the data and enhance medical imaging?
Okay, so that's again a very good question, and this points to the fact that garbage in equals garbage out, right? So when you use your images, or for that matter, when you use any sort of data to develop any models, don't just jump into training of deep learning algorithms. Don't just jump into the best possible algorithm that's out there or the latest algorithm that's out there and start training your model. So always try to understand the data first. And this is again what I try to tell all my students in my lab. So it's understanding the data and curating the data is probably 60 to 70% of your job done. So by that, what I mean is there are different ways in which you can dissect your data. You can look at, for example, different kinds of representations coming from that data, and you can, let's say, perform, or you can cluster them using techniques like TSNE, for example, and you can observe the local clusters that emerge, and then you can go back to the data and see, hey, I see that there's a cluster of 5 patients there. What's going on with that particular cluster? So you try to understand this after you analyze this using different methods and try to see what's going on inherently with these images. So we have actually developed an approach for for digital pathology, where we look at these images, we pass them through an algorithm that we have developed, and it comes up with a score, a quality score, and that quality score tells us if that image is suitable for downstream analysis or not. So in one particular project, no matter whatever algorithm we used, there were a few slides which always ended up being misclassified, and we had not really done a quality check for that. But when we went back and we looked at the slide, one of those slides, these were glass slides that had been stained with, tissue that had been stained with H&E and then mounted on a glass slide. We found that a couple of these slides had cracks, and a few of these slides had strands of hair in them. And obviously, no matter what we did, we always ended up with bad results. So again, what we learned from this experiment is that always look at the data first. You can look at different graph-based approaches. You can extract different kinds of features, be it texture features. You can extract different handcrafted features, or you can even do representation learning. For example, self-supervised SSL-based features, self-supervised learning-based features, and then plot out these features and then see if you have some anomalies that emerge. And that is one way of addressing quality of data before you start training any algorithms.
Thank you, sir. So another question which we have is, shouldn't we focus more on data collection rather than data manipulation, because the main fuel for neural networks is accurate data sets?
Short answer is yes, if you can. Not all of us have the liberty of collecting our own data. So if you are in a lab, or if you have the opportunity to collaborate with a lab that collects data, by all means go for it. So understand how the data is collected, understand the different vagaries of collecting data sets, and how even slight tweaking of certain parameters can completely change the signal to noise ratio, for example, right? So if you can understand, if you have the opportunity to do that, definitely do it. But not many of us who work in the field of medical image analysis have that opportunity. So if you do, that's great.
Thank you, sir. I've seen somebody raise their hands.
Okay, so
Abranta,
yeah, go ahead.
Hello, sir. So you talked about using autoencoder based networks for generating newer images in case of shortage of data. But autoencoders based networks, like, they don't generate newer images, right? To generate newer images will require variational autoencoder based networks. But then those networks, means VL based networks, those will end up producing very fuzzy images. I've tried out in some human faces and tried out different types of training, and they produced very fuzzy images. And GAN based network trainings are very difficult and prone to mode collapse. So what was the technique that you used, or what types of training procedures you followed for those?
Yeah. Okay, so apologies if I wasn't really clear about the method. You can actually learn more about that method in a recent Mikhai paper, long swapping autoencoder. So what was done there was the autoencoder was used to learn representations. So we had this texture encoder and the structure encoder. So the texture encoder learned the structure of these images, and the texture encoder learned the texture of these images. So we use the autoencoders to actually learn the structure and the texture, and then we did swapping of the structure and the texture to generate new images. So yeah, so the autoencoder itself was not used to generate images. So it was used to learn the representations of or to disentangle the structure and the texture representations of these images.
Okay, so you first mathematically derived out the structure and the structure representation of the image and the texture representation, and then trained the autoencoder to basically disentangle the multiple levels of features, right?
That's right. Yes.
Okay, got it.
Thank you, sir. So thank you everyone for all of your questions. So all great. I hope that the ideas and views shared during this session paved the way for sustained fruitful result. Now, all great things must come to an end, and unfortunately, so must this wonderful session on medical image analysis. Thank you, sir, for an informative and interactive session. In a world where medical imaging techniques are growing exponentially and never cease to amaze us, I'm certain that your words have been an inspiration to all the innovative minds out there. With that in mind, it would be my honor to call upon Ms. Sneha Swain, Vice President Genesys, onto the platform for the vote of thanks. Thank you, everyone, and hope you had a great session. On to you, Ms. Sneha.
Thank you, Abhijit. Good morning, everyone. I am Sneha Swain, the Vice President of Genesys, and it is an honor to be a part of conferences that highlight these topics which are popular yet shallow understood. I thank Dr. Prateek for his insightful session on AI and biomedical image analysis. Sir, we appreciate your time and efforts in helping us make this session a success. I also thank Professor Khalid Beg, our students and participants for being with us all throughout the session, and hope that you have a list of takeaways as well. I also thank our team members from Genesys who left no stones unturned to make this session an effective one. We also hope to see you in the upcoming sessions for today, and definitely attend the closing ceremony to know whose idea captured the judge's attention. Thank you once again and have a great day ahead.
Thanks again, everyone, and thank you for the invitation. And if you have any questions, feel free to reach out to me at the email on your screen right now. And yeah, I'm also actively looking for collaborations. I'm actively looking for interns, students. So yeah, feel free to reach out to me. Thank you. Thanks again.
Thank you, sir.